Good afternoon, one and all. On behalf of Gokhale Education Society, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, Nashik, I, Mrs. Rashmi Vishal, Assistant Professor and In charge HOD, DPharm, extend a warm welcome to you all to the third session of fourth day of the one-week international e-FDP, e-STP on recent advancements in pharmaceuticals, academic, research, ethics, and social perspective under the aegis of Internal Quality Assurance Cell. The speaker for this session is Dr. Chinar Pathak. It is my privilege to introduce this eminent personality to you all. Dr. Chinar Pathak is currently working as Scientist 2 with Ming Therapeutics, Cambridge, UK from 2022. He graduated in pharmacy from North Maharashtra University, Jalgaon in 2005. He completed his master's in biotechnology from VIT University in 2007. After his post-graduation, he joined Piramal Life Sciences, India as a research associate in R&D Center and worked there till 2008. In 2009, he joined Seoul National University, South Korea, and was awarded PhD in Physical Pharmacy, Structural Biology in 2014. He has also completed 2 postdoctoral fellowships from Seoul National University, South Korea in 2017 and University of Leeds, UK in 2019. Dr. Chinar Pathak has also completed 2 certificate courses in HMX Pro Immuno-oncology and HMX Fundamentals Immunology from Harvard Medical School and Mental Health Workplace Responder from St. John Ambulance Future Calls. He was awarded Level 2 award in 2023. Dr. Pathak then joined University of Leicester, UK as a research associate, postdoctoral researcher and worked there till 2022. Since 2022, he is working with Ming Therapeutics, Cambridge, UK as Scientist 2. Since 2008, he is involved in team collaboration, guidance and mentoring. Dr. Pathak has more than 15 years of experience in structure-based drug discovery and drug design, antibody discovery, protein engineering, construct design, protein production, expression and stabilization, protein 3D structure determination by X-ray crystallography, NMR, functional assays, molecular interaction, and dynamics. He is also the proud recipient of 2 lifetime awards, AFHEA Associate Fellow of the Higher Education Academy in recognition of his teaching and mentoring. He received this award in 2020, and he is also the proud recipient of the Korean Government Scholarship Award, which is internationally awarded to only a very few candidates for PhD. He has also received the Young Scientist Award, a prestigious and competitive award by the Korean Protein and Peptide Society in 2014. He has received research grant as co-PI under Indo-Korea Joint Program of Cooperation in Science and Technology from 2014 to 17. His featured publication was selected by Rigaco and has been published in the Crystallographic Times, Volume 4 in 2013. He's also a national level chess player who has played 1 national game in 2006 and 7 times played in the state chess championships from 2000 to 2007. He was invited as speaker at Asia Pacific Nuclear Magnetic Resonance Symposium, India in 2017 and delivered distinguished lecture at 18th Korean Peptide Protein Society Symposium, Korea in 2014. Dr. Pathak has to his credit about 12 research review paper publications in various national and international journals of repute. His H-index score is 6, I-10 index 3 with more than 84 citations. Dr. Chinar Pathak is going to deliver his expert talk today on the topic targeting proteins from immuno-oncology conditions to develop new drugs. Welcome, sir. I now hand over the session to Dr. Chinar Pathak. Thank you.
Thank you, madam, for that introduction. I wasn't expecting such a long introduction, but thank you all. It was a pleasure talking to you today. And in today's session, I would like to give a flavor of what I have been doing or involved in during my time in South Korea and in UK, especially in Leeds, Leicester, and currently in Cambridge. Today I'm going to talk about the proteins that we get from the disease conditions, especially the disease that are related to immunology and cancer, and how we can use those conditions, how we understand them to develop the new drugs. Can you see my slide transition? Can anybody just confirm?
Yes, yes.
Okay, great. Thanks.
Thank you.
So can you go into presentation mode? In slideshow, I mean.
Please go into the slideshow mode.
Yeah. Yeah. Now?
No, sir, actually, it's not visible. Sir, have you shared your entire screen?
No.
Yeah, so can you do that so that we can see your slides in the slideshow mode then?
Oh, I'm sorry for that.
No problem, sir.
Yeah. Can you see my slides now? And the transition?
Yes, sir.
Yes,
sir.
yes.
Okay. My apologies all for this old guy to be a technical savvy. But as I was saying, my research from past 10-15 years has been with the different disease conditions and the proteins that relate to these conditions. So today I will talk about amyloids. So these are part of beta 2 microglobulins, or beta 2M as they call. This is part of MSC class 1 that causes disease related amyloidosis, and it results in the failure of kidneys. The seconds are pathogenic bacteria, especially Helicobacter pylori, that occurs in the stomach. Nearly 80% of the population have this organism in our stomach. Currently, the people who are listening may have this organism in them. I have it. So it causes oncology, and it is responsible for antibiotic resistance and enzymes. So these enzymes are specially related to blood, heme peroxidases. So they are responsible to carry out the redox reactions, and they are responsible for multiple numerous diseases throughout the body. So when we talk about the amyloids, uh, here we can see that the proteins which are shown here are transition from unfolded to the folded state, but there are numerous pathways. These pathways are certainly known in the field. You can find them in your biology, biochemistry, and immunology textbooks. But what we don't know that how do they fold, and during this folding, sometimes they become misfolded. That results to be aggregation prone conditions, and that results into amyloid fibrils. Now, MHC class 1 is a part of our immunity system where alpha 1, alpha 2, and alpha 3 proteins gather together along with beta 2 microglobulins. And upon unfolding to the folding states, it goes through a transition called intermediates, and the transition of proline 32. So this amino acids is responsible for the correct folding of this protein. But proline, as some of you may know, have a pentameric structures. It doesn't have one NH in its C alpha positions. That relates to carrying of delta N6 variant of this protein, and the serum concentration becomes about 20 times high, and that relates to dialysis related amyloidosis. So when I was working on this protein, there is a new group adjacent to my laboratory. We're working on some protein aptamers. They call them as affamers. This is their patented technology. What these proteins are, they are very inexpensive. They are less complicated compared to antibodies. They can be developed much quicker, stable, and they can be easily adapted for variety of use. So the idea that pops us in our group, in our mind, is how we can use these protein aptamers to create a drug candidate for these kidney failure diseases. So a little bit background about my protein preparation. So we used normal PET systems, and I created all these different variants of the proteins. So full length protein is about 98 or 100 amino acids, and the delta N6 variants is 94 amino acids, and we created different mutations of it. with his-TREP-Q anion column. So these are cation anion exchanges, ion exchanges columns. We purified the protein. We use the inclusion body formations because by that way we can get a huge amount of protein in a small culture, which is beneficial for time saving, money saving, and it will readily help in the research project. Then we polished our protein using SEC column. This is size exclusion chromatography, and we verified it with tandem mass or ESI mass spectroscopy. Then I created two variants called serine 33 cysteine and isoleucine 92 cysteine in delta N6 variant of this protein. We changed that amino acids to the cysteine to make it biotin melamide formations. We purified this biomolecule to create a biotin melamide conjugate of this variant. With this conjugate, we screen the phase display library that we had in the hand. So phase display library is about 10 power 10 affimers. So that is about more than 1 billion proteins together in a test tubes. We use bacteriophages. So these are the viruses that infect the bacteria. And with the functional assays that I have shown here, this is called panning rounds in a phase screening displays. So what panning rounds are? You mixed your drug candidate or this panel of candidates, which are 1 billion compounds with a protein. You wash it, then you rescreen it, and you repeat this process for 3 times. And at the end, you get the high binders to these proteins. The affinity might range at least nanomolar to sometimes picomolar or even femtomolars. So biotinylated S33C, the structure is shown on the left, which we adapted to a nickel to the streptavidin plate. So these streptavidin plates will attach to the biotin and the protein will be flagged outside of it. The reason for the two formations were we can change the orientation of the protein in all. Then we collaborated with Darren Tomlinson, who is a professor, very knowledgeable and very good professor at University of the Leeds. So he carried out the sequences and these are the ranges for it. Then the main problem was phase display library yielded 28 strong affimers and question is which one is the best. So I developed a method which is called histidine fluorometric assays. So what I did was I mutated a residue to add a cysteine. Then I used ATO 488 melamide conjugate and I purified the protein. And on the other hand, these affimers were cis tags. So hexahistidine tag or octahistidine tag in the protein sciences is quite common. So we can use nickel NTA coated surface. I specially used the 96 well plate for this for high throughput screening. Then we added the ATO 48 melamide conjugate over it and we just simply wash it. After washing, the idea is the best candidate can give the fluorescence at 488 nanometer and we can check it. So this is the setup. Idea is the best candidate or the highest affinity candidate will give high fluorescence and the lower or the low binding candidates will give low fluorescence. On the right hand side is the raw data. Once we translate that everything into the graph where a human eye or where computational programs can come to the hand, we checked it. So this gives us the increased sensitivity and specificity. It also helped us in getting rid of non-specific binders. So we performed this assay on 2 different variants of 3 proteins and at 2 different pH. One pH 6.2, which is disease prone because the kidney or the dialysis related amyloidosis disease, the location in the human body for that disease is about pH 6, and 7.5 is close to the physiological relevant pH because the blood is 7.4. With all this data, we created the different affimers for it. For the sake of preserving the sequences, we call them affimer 1, 2, 3 and the numbers to preserve our intellectual rights for it. Now, we purified these affimers with nickel NTA beads and we did the SDS page. Again, mass analysis comes handy when we see the protein is about 99% or even more in its pure form. Then I did kinetics. Just a minute, please. Sorry. Yes, it was here. Sorry, that's okay. That's okay. Thanks, Eleni. Yeah. Sorry for the interruption, guys. That was director of our facility. So they just wanted to have a quick chat with me. So back to presentation. Um, so I was talking about how we did the functional assays. So this is called THT assay or thioflavin T assay, where we can see the protein is making fibrils. So in simple words, fibril is a form of protein binds to itself and make a long fibers. So monomeric protein can make multimers. So these multi number can range anything from 3 to 30 or 300. And these long fibers makes the diseases. What happens is whenever the protein makes fibrils, the thioflavin T will bind to it and give increase in the fluorescence. So this is a quick and good assay where we can see the delta N6, the native protein is in black color. Mouse beta 2M, which is a variant of the same protein in mouse, is shown in red, and the true drug candidates. So the disease that can make in a human body at a physiological conditions is happening is around 10 hours. However, the addition of affimers make it longer, and even after 100 hours we cannot see the formation of fibrils. That means these two affimers are prohibiting or limiting the disease progression state. So we tested it in two different conditions, de novo, which resemble to the early stage or early occurrence in the human body, and 3% seeded experimental in vitro condition that is similar to the advanced stage disease in human body. We tested them with negative EM, this is electron microscope images. We can see the fibril formation in black that happens with delta N6 in the body. But with addition of affimers, which is shown in pink and blue, we can see there are no aggregation of this protein or there are no fibril formations. Then we went ahead and tested everything by NMR. So this is two-dimensional NMR spectroscopy, and the protein was labeled 15N. So nitrogen is labeled with 15N isotope labeled amino acids. So the effect of affimer, we can see with increasing amount of them, and we titrated the KD. The KD belongs to 8 micromolar. Delta N6, beta 2M, and the affimer, we mapped the residues on both of them, and the interacting residues are shown here. Then we wanted to see whether these sequences are responsible for it or not. So we created a chimera of this protein. So the chimeras are the combination of mouse and human beta 2M part of MHC class 1s. Then we made the chimeras based on our predictions. Again, we purified with common techniques that I've shown before, and the chimera activity has been tested. In conclusion, we came to know that mouse beta 2 interaction on delta N6 does not inhibit self fibril formations. However, it does delay. It's important because mouse beta 2M until today was known as a, natural or known as the best candidate to inhibit fibrils in humans. However, with our studies, we tested its efficacy with three different proteins, alpha-synuclein, A beta 40, 42. So these are three different proteins occur somewhere else in the body, and they're related to the different multiple diseases. I'm not going to talk about these proteins, but these are considered as competitive or positive controls in my experiments. So we came to know that these affimers are very specific to only delta N6. Then we collaborated with Patricia Mann, and we did in vivo studies using C. elegans, because C. elegans are closely related to the human genome. So in normal, this is the how the C. elegans move when they are happy and in normal healthy conditions. When we injected delta N6 phenotype to them, we can see the movement is stopped. We measure that in something called BPPS, that is body per second movements, and we can see that worms are being diseased, they are not moving. After injection of affimers, the worms has started moving, indicating that the disease has completely cured and the worms becomes healthy again. So the significant finding is for this project was we created an assay which is nowhere in the literature till today. We discovered three potential drugs, and the affinity for these drugs is 10 times better than any other marketed product today. Right. So I will switch a gear a little bit, and now I will talk about antibiotic resistance and virulence in toxin antitoxin systems. So these are the part of Helicobacter pylori and oncogenic protein. So quick bit of history. So pathophysiology of Helicobacter pylori is known from past 20 years. Two people, Warren and Marshall, has already got a PhD Nobel for it. When I was studying this, only two systems called HP0894. So HP belongs to Helicobacter pylori. So 9495 is toxin antitoxin system, and 9293 another toxin antitoxin systems are known. So these were only two systems known at the time, and both of them were discovered in our laboratory. What happens is 94 toxin is always located downstream in our genome, and 95 antitoxin is upstream of it. We can test them with the pull down assays and RNA sync assays. So I took this protein under my investigation. I did cloning, protein expression. We made crystals of it, and we take these crystals to the Japan. So this study was done in South Korea when I was a PhD candidate there. So we took these crystals to the Japan, who had a photon factory, synchrotron crystallization, or sorry, a synchrotron technique that will help to see the structure of the protein. So we all know that carbon-carbon bond is 1.56 angstrom. So anything close to that will give us a native resolution for the protein. However, the molecular replacement did not work. So I did selenomethionine labeling. So selenomethionine is this amino acid. I'm showing it on the structure. But however, we found it one unintentional mutations on the 66th residue. Then we reinvestigated in why it's happening, and we came to know that this is stress-induced mutagenesis. It made much more sense when we saw the protein again, because it's a toxic protein, and this DNA sequencing analysis confirmed that it's a combination of native as well as L66X mutant. To overcome this issue, I expressed this protein with intense. So I used the N-terminal intain, where the protein was attached to the CBD, which is chitin binding domain. I purified it with chitin column, uh, got higher purity using cation exchangers, and we took crystals back to Japan at Springate this time, and we got a beautiful diffraction of it. With this, I came up with 1.2 angstrom native resolution. This was my early days, and when I found that the electron revolving near any of the amino acids, we can see its electron density, which is blue in color, and it was really beautiful at the time. So this was native 0894 resolutions. Then with the homology modeling, I came to know that these three histidines and are arranged in a specific way. With the structural analysis, or what we call as DALI scores, we came to know that YFQ, a part of E. coli system, Rayleigh system, and RNS-T1 are structurally similar to Helicobacter pylori 94. With this data, really being being a part of a metal interacting domain, I created a functional assay where I added the mRNA of my protein along with different metal ions and find it whether the protein degradation occurs or not. In a simple words, we can call it as a ribonuclease test, and the significant structure made copper as an identified cofactor, because in presence of copper and zinc, the protein degradation didn't occur. That means somehow this copper and zinc are stabilizing the protein structure. Then because copper was identified, I did copper bound structure of it. In a simple way, I mixed ligand, which is copper. I equilibrated the protein. I carried out the crystal conditions, optimize whole this screening conditions, which is buffer optimization and cryosolution optimization, and we got a crystal using acetate, citrate, and PEG conditions, and we took the structure. So we can see that one of the uh, copper is located in between two monomers of the protein. In detail, I see the reorientation of histidine 84. So the green color is apoform and the blue color is copper bound form. And this reorientation was more than 7 angstrom, which was quite remarkable. Because of this reorientation of these residues, we can see a triad forming by histidine 84, histidine 60, histidine 47, and all this was stabilized using glutamate 58, which is again negatively charged amino acid. Same I carried out with the HP0892, the second antitoxin system in Helicobacter pylori. So we did cloning, and I purified the protein with nickel NT, because the protein was labeled with hexahistidine tag at its C terminal. Then I used thrombin cleavage site to take out the histidine, and I purified with sec columns, the gel filtration chromatography, and the purity was judged to be more than 99% pure form of the protein. Then I did thermodynamics assay. This is a circular dichroism where we can see the melting temperature or melting point of a protein. The apoform is shown in blue, however, shown in black, which is just behind my pointer, but the melting temperature was increased in presence of zinc. So we can see that with presence of zinc, we got 72 degree, which is 10 degree higher than the apoform of the protein. That triggered me to do the titration with this zinc, and this is ITC, so isothermal calorimetric assays, where I see each monomer of the protein has two binding sites. That means it requires two molecules of the zinc atom. We also see the enthalpy and entropy with this. And with that, I did the metal binding analysis, and sorry about that. I mixed metal with a protein, did the crystallization trials, and we got a beautiful structure at 1.8 angstrom. At this time, I used molecular replacement, and quickly we ended up with the structure of the 892 toxin with zinc bound form. Strikingly here, we also saw the involvement of three histidines and one glutamate, shown here as histidine 60, histidine 47, histidine 86, and glutamate. So again, we saw the triad forming in the toxin form of this protein. So we took the help of NMR. So now this is three-dimensional NMR. So I labeled carbon and nitrogen using 13C and 15N isotopes. And we collected the 3D data set. Then I labeled the whole protein. I determined the structure, and we used that and projected here on 2D monograph so that human eye can see it better. So apoform of the protein and increasing concentration of the zinc is shown. The ratio is shown with the corresponding color. And we can see that the location of these two zincs are on the beta sheet face of the protein. Tracing it back, so I mutated this protein, purified it again, and crystallized it. With this, we got the native form of the protein. We got the catalytic residue histidine 86 because reorientation was more than 7 angstrom. Only one basic residue, glutamate, and a combination of them. So I mutated on both of them, all three of them, and we did ITC again to see how much effect they have on each, or how much effect they do have in the RNase activity. With the native form, we see the threefold decrease with histidine 86A, but completely abolishment of activity because glutamate was the only one base residue in these three positive histidines. A combination of that again resulted in abolishment of activity. The number of sites and the enthalpy change is shown in the table. With this apo and metal bound form of two proteins, I overlaid them together, and we can see the similarities between the two structures as well as the pocket that has been made by these toxin antitoxin systems. Then we took the help of bioinformatics, and we came up with this. We actually use this server which is called CAST-P. So it is modeling and computation, and it came up with this deep cleft site. So deep cleft site is nothing but the putative drug binding site, and we can use this drug binding site to see which drug can be capable to interact in here. For the patent and IPR analysis, I'm not showing our drug candidates because obviously for patent issues I cannot show what drugs or what drug candidate I'm using in this project. This is extra diffraction analysis and refinement statistics. In normal words or simple words, how good or bad the study or the structure is. Anything about 20% of R work and R free is considered as good. So anything less than 20% is considered really good. So with some proud moments, I can say that the structures were deposited to the PDB server, which is available freely to everyone. We contributed to the toxin endotoxin system field considerably. With that, I will go to my third and last topic, which is heme peroxidases enzymes. So these enzymes occur into the blood, or heme peroxidases are the enzymes that bind with the heme. So I took two enzymes, one from soybean, it's called APX, soybean ascorbate peroxidase, and second is cytochrome C peroxidase from the human body, CCP. So, in a general form, what we know until the study was the enzyme and its photoreduction can happen by two states. It's called compound 1 and compound 2. So, these proteins do contain heme. They are distinct in their structural properties. They are expressed in all kingdoms of life, whether you take plant, animal, human, bacteria, fungi, amoeba, or anything about that. Then we can difference. We can see that the difference was in the bond length in the compound 1 and compound 2. So these numbers include, this number indicate the bond length between the heme and the oxygen, so the redox reaction state of the protein. However, we can see that multiple different papers or book chapters indicate different numbers, so there is ambiguity. And this is the issue of photoreduction. So whenever we see a structure in paper or books, that means the structure has been determined either by NMR or X-ray or cryo-EM. However, there is this ambiguity. This ambiguity will result in making problems or finding the drugs. So we took the task under our study and see what is the actual length and how we can eliminate that. The difference may also arise from photoreduction. So it is one of the physics phenomenon. In a simple words, when the X-ray shoot a protein structure, it creates a heat. That heat may disturb the local environment of that amino acid or protein, and that photoreduction, the whole process is known as photoreduction, and this will change the bond length. Even by a slight change, even by a minute change, these numbers will change. Obviously, if the bond length changes, the pocket changes, and the drug development process delayed. So we wanted to see how they will work. The problems that we had in our hand at the moment is heme is very sensitive to redox, and the ligand state changes can be solvated even at the cryo temperature. So cryo temperature is minus 196 degrees Celsius, and how we come over that, I will just come to that in a second. So we used XFEL. So XFEL is relatively new technology. It is commonly called as fourth generation synchrotron. And in a short graphical abstract, I can explain that we can see the horse at its resting state or its running state. However, whenever the horse runs, there is a movement of these legs. So in a more scientific way, I can relate that to the enzymes. So enzyme along with the substrate results in a product, but this transition may go through number of steps, and XWELS is the only technology or only technique that can take photographs of this transition, so we can see the movement of electron in real time. So this real time is picoseconds, and it takes thousands and thousands of photographs within a very short span of time. So traditional microscopy is done with synchrotron. It's done at 100 Kelvin, and we can get the resting state of enzymes or bound state or the product state. The problem is it lacks dynamics, and the 90% of the structures which are deposited in PDB per year lack this dynamics because it's a static structure. That makes sense, right? The second technique is cryo-EM. So cryo-EM is done at liquid, done with liquid nitrogen. So again, we go to the low temperature. We do get microsecond or millisecond time resolutions, and this complements crystallography. So the combination of those two is really good for the field. However, we do get class averages, and there is a limited dynamics. This serial crystallography comes handy when the entire reaction can be done at a room temperature, because obviously the human body or any animal body lives at room temperature. We don't live in liquid nitrogen, don't we? So this XFEL, they can go in femtosecond pulses. So when we go in femtoseconds, 10 power minus 15, we can see the bond vibrations. Also, this is non-radiation induced technique. So that means we don't see any damages. This is microsecond time resolution. We can use different mixing strategies to optimize it. We can get microcrystal slurries. This point is especially important because in my previous slides, I mentioned that we optimize the crystals or the conditions. So that was one sentence I said, but optimizing crystal condition, it takes months or even years. So I have spent about 2 years in optimizing one crystal conditions. So that was one of the special times. However, in this Excel, we can avoid all that limitations. So we get quick and we save a lot of time and energy optimizing the crystal conditions. Also, we can see the electron transfer mechanisms. So we can see from how the enzyme is converting substrate to the product, and we can see the different stages of it. So again, I cloned the protein in two different constructs. I purified them with nickel NTA. I polished the purifications using sec columns, and I did the initial. We got the initial hits using crystallizations. I optimized it just a little bit to make the crystal bigger. With time, we end up with good red colored crystals, which are shown here and here. So these are kind of long needles or almost cube-shaped crystals. Then we went to SECLA. I mentioned that synchrotron. So this is a photo of synchrotron. This is located in Springate, Japan. The work was done here in Leicester, UK. So we took a flight to the Japan, and it was quite fun to travel there because it's in the middle of nowhere. Because this crystallization synchrotron needs nuclear power energy, and it should be or it must be made where normal public or population will not be affected by the nuclear radiation. So this is synchrotron. It's called third generation, and each one of these points where I'm showing my pointer can sit and analyze your crystals and get the structure. So most of this synchrotron, there are only 12 in the entire world, and each one of them with this ring will have about 30 to 40, of course it depends on the size, but about 30 different outposts with each one of them. What I'm showing here is this straight thing, this straight line, which is called as SACLA. So the radiation is located at one end and it bombards the beam of electron up to the next end, and this small outpost is the where we will do the experiments. With this setup, we came up with the APX and CCP crystals, and the pocket is shown here. And this is the kind of heme. So heme is kind of heme looks like cyanocobalamin, and it's a planar structure. So it sits in the middle, and we were able to distinguish the binding pocket of the heme and its different electron transfer mechanisms. With the time constraints, I did not show all the electron mechanisms here because it would take hours to explain. In short photograph, we can see that CCP compound 2 and SAPX compound 2, we were able to distinguish and say with confirmation that CCP, cytochrome C peroxidases, has a state called compound 2 with a bond length of 1.76 angstrom, and we can see the interpretation of this result. Now, ascorbate peroxidases has the bond length of 1.87 with a resolution of 1.50. With CCP, the resolution was 1.0. So what we found out with this is a damage-free structure used obtained by XFEL. The location of individual hydrogen atoms and protonation states was found out. We can use this data to design the new catalyst. New catalyst will obviously will be helpful to make the new drug candidates for the protein, right? So with that, I came up with the end of my talk, and I would like to answer any of the questions that you may have. Thanks.
Delegates, the session is now open for question answers. If you have any questions, doubts, please post it in the comment.
Should I stop sharing my screen?
Yes.
Okay.
Okay.
Okay. Thank you.
Yeah. Well, thank you all again for giving me your time and opportunity to present my research. It was lovely seeing you all. Thanks. Bye.
Thank you. Bye.
Bye.
The session is ended now.
Oh.